There are probably fewer atoms on Earth than possible human faces. Picture every atom on the planet gathered into a single line. Every grain of sand, every breath of air, every living cell. All their atoms combined are dwarfed by the faces you could create. Now, compare that line to just one feature. The shape of a single eye. Round, almond, hooded. 3 variations from one tiny feature. Multiply by every possible spacing between those eyes. Narrow, medium, wide. Each eye shape now has 3 spacing options. The count triples in a single step. Then multiply again by every possible nose width and lip curve. Wide, narrow, flat for the nose. Thin, full, wide for the mouth. The branches keep splitting, each choice multiplying everything before it. Layer in skin texture, pore patterns, and hair direction. Smooth, poured, rough. Straight, wavy, curly. Each new dimension multiplies everything that came before it. The resulting space already dwarfs the atoms that ever existed on Earth. The tiny sphere on the left, all those atoms, vanishes compared to the branching tree of possibilities on the right. Repetition is not merely unlikely, it is statistically implausible. Across all of human history, no 2 faces ever need to be exactly the same. Thousands of genes act together before any face forms. 3 key players, PAX3, DCHS2, and EDAR, already shape the facial landscape from the very start. Take PAX3. This single gene influences the width of facial bones. Even a subtle variation here can shift the entire skeletal frame. Small changes in PAX3 already create distinct bone outcomes. 2 alternate versions diverge toward different facial structures. Now, DCHS2 activates, adding an independent layer. It controls skin tension and texture, completely separate from the bone foundation. Each added gene multiplies the possibilities. Bone variants and skin variants already combine into dozens of potential starting configurations. EDAR goes further, influencing hair follicles and brow ridges. A third independent axis of variation enters the mix. 3 genes. Each carries multiple variants. Already, thousands of distinct starting states emerge, well before a face is even recognizable. Neural crest cells begin their journey from the neural tube, carrying the genes that will eventually shape facial features. Timing differences already send cells along diverging routes, creating the first layer of developmental variation. Random delays push some cells to arrive later than others, injecting stochastic noise into the developmental program. Epigenetic marks, like methylation tags, silence or activate genes within those cells, adding another layer of regulation. As migration completes, cells settle into different tissue regions, forebrain, midface, and jaw, producing divergent outcomes from the same starting genome. Each added event, delays, methylation, variable migration paths, compounds the facial differences already forming during development. The same genes now produce visibly distinct tissue layouts. Development itself is a source of individual variation. Even identical twins start from the exact same genetic blueprint. 2 embryos carrying perfectly identical DNA. But the journey to unique faces has already begun. Stochastic timing is the first crack in the mold. Cell division schedules naturally drift apart. 1 twin cells arrive slightly late, nudging facial development off its initial course. Epigenetic marks add another layer of divergence. Chemical tags, methylation groups, silence certain genes in 1 twin, but not the other, subtly changing how local tissue grows and forms. By adolescence, those tiny early differences have snowballed. Bone structure, skin texture, subtle feature shifts. The faces are already measurably distinct, even to a casual observer. In adulthood, the human visual system becomes a facial difference detector. We automatically scan and recognize these unique configurations, often without conscious thought. Evolution tuned our brains for exactly this task. Social recognition was a survival advantage. Spotting who is who in your group, friend or stranger, mattered deeply. Identical genes, unmistakably unique faces, even twins who share the same DNA code end up as visually distinct individuals. No two faces remain truly identical. The opening comparison returns. Atoms on Earth versus every possible face. Those same impossible odds. Genes laid a foundation. Each new combination of ancestry cranks the probability wider before development begins. Then, developmental noise adds its own randomness. Neural crest migration, epigenetic marks, microscopic chaos that makes every face a one-off. Even identical twins diverge. Same DNA, different facial geometry. The system guarantees uniqueness. And our brains are wired to catch it. We distinguish faces at a glance, sensitivity that proves the differences are real. Across all humans who have ever lived, across every culture and century, the repeat slot has never been filled. No two faces have ever repeated in history.